Eukaryotic DNA is organized into nucleosome structures, and this complicates replication. This isn't true in prokaryotes, which really don't have histone structures and don't have nucleosomes. Eukaryotic nucleosomes are composed of a specific mix of histones. There are H2A, H2B dimers, and there are H3, H4 tetramers. During DNA replication, histones have to replicate too. And that's complicated because you have to have a precise nucleosome structure after replication, replacing nucleosomes as you move along the replication fork. So the cells have to synthesize new histones in very specific amounts because there is a specific ratio of H2A, H2B, H3, and H4. So the regulation of their production is very important. You have to have the ability to meet the demand for creating a complete replicate of not just the DNA, but the package structure of the DNA. The amount of histone produced is regulated by the level of histone mRNA. Degradation of the mRNA determines amounts of each of the histone monomers that are produced. If we look at what's happening at the replication fork, you'll see that the H2A, H2B dimers are released where the fork opens, and the H3, H4 tetramers are randomly staying with one of the separated strands. There seems to be this mode of dissociation that differs between H2A, H2B, and H3, H4. The dogma has been that the old H3, H4 randomly goes with one or the other of the daughter complex strands, and the old H2A, H2B are released and mix randomly with new ones to form the new chromosomes. The new H3, H4 that associate with the open sites where they had been released are modified to match the pattern of the old H3, H4, irrespective of which strand the H3, H4 is associated with. The systematic way of describing this is that old and new histones reassemble in a way that preserves chromatin structure on both strands of the DNA with a little help from modifying enzymes. There's a newer idea that it is the modifying enzymes that are retained with the DNA, and they modify the newly synthesized histones to match the original pattern. There is still some controversy about precisely how it happens that histones and the daughter molecules of a DNA replication process end up being identical. How is it that the histone modification pattern is replicated? Because it's very clear that that pattern is replicated, and that it can be a form of significant variation between cells and between individuals. Replicating the histone modifications is important because H3 and H4 are particularly sites of modification. And as we've said, there are reader-writer enzymes that in some way recognize and duplicate the modifications to match the parental H3, H4. So if we start with the parental nucleosomes, after replication, only half of the daughter nucleosomes would have modified histones. So these modifying enzymes replicate the parental pattern of modification so that they have exactly the same modifications when replication is completed. This preservation to maintain precisely the histone modifications is one mechanism by which epigenetic changes endure and can be passed on. Because remember that the nature of chromatin structure, which is determined by the nature of the histones and how they are modified, is a factor that determines how that DNA is expressed or not. These epigenetic changes are not changes in the base pair sequence. They are not genetic changes. They are not part of the DNA sequence. But they do impact the expression of specific regions of DNA in ways that can be inherited from cell to cell and from generation to generation. The same nucleosome, depending on its modification, can cause a repressed state, an active state, or bivalent state so that the DNA can be responsive to other signals from the cell. What types of environmental impacts could you imagine that would impact histone modification, and why? It's very clear that there are environmental impacts that affect histone modification, that are engaged with epigenetic changes. So how might that happen? And would you anticipate that methylation of histones would alter nucleosome structure more than acetylation? Why or why not?